Before we begin, welcome to Inanna Board Review, designed for medical laboratory professionals preparing for CLS, MLS, and board certification success. Inanna Board Review simplifies complex laboratory concepts into easy, high-yield learning designed to help you understand, analyze, and excel. In this video, we will begin our introduction to proteins, essential biomolecules that play critical roles in structure, transport, regulation, enzymatic activity, and many other vital functions within the human body. Let's get started. Overview of total proteins. Proteins are one of the most important biomolecules in the human body. They are large macromolecules made of amino acids linked together by peptide bonds. Proteins perform many essential functions including structural support, enzymatic activity, regulation, transport, defense, and movement. In clinical chemistry, total protein measurement is commonly used to evaluate nutritional status, liver function, kidney disease, and inflammatory conditions. Most total protein measurements are performed using serum or plasma samples. Protein structure. Proteins have a hierarchical structural organization that determines their stability and function. The primary structure is the linear sequence of amino acids linked by peptide bonds. The secondary structure refers to local folding patterns such as the alpha helix and beta sheet, stabilized mainly by hydrogen bonding. The tertiary structure represents the overall three-dimensional folding of a single polypeptide chain. Finally, quaternary structure occurs when multiple polypeptide subunits combine to form a functional protein, such as hemoglobin. Classification of proteins. Proteins can be classified according to their structure and composition. Simple proteins are composed entirely of amino acids. These may be globular proteins, which are soluble and functional, or fibrous proteins, which provide structural support. Conjugated proteins contain both a protein portion and a non-protein component called a prosthetic group. Examples include glycoproteins, lipoproteins, nucleoproteins, and metalloproteins. This classification helps explain the biological and clinical roles of proteins. Absorption spectra of proteins. Proteins absorb ultraviolet light mainly because of aromatic amino acids, especially tryptophan and tyrosine. The maximum absorbance typically occurs at 280 nanometers. This property is widely used in laboratories to estimate protein concentration using spectrophotometry. Absorbance at 280 nanometers is a rapid and commonly used method for protein quantification. Amino acid properties and dipolar nature. Amino acids exist as zwitterions, also called dipolar ions. This means they contain both a positive charge on the amino group and a negative charge on the carboxyl group. The net charge of an amino acid changes depending on the pH. The isoelectric point, or pI, is the pH at which the amino acid has no net charge. This property is important in protein separation techniques and electrophoresis. Overview of peptide bond formation. Peptide bonds are formed through a dehydration or condensation reaction. During this process, the carboxyl group of one amino acid reacts with the amino group of another amino acid. A molecule of water is removed, forming a covalent amide bond called a peptide bond. Repeated peptide bond formation creates polypeptide chains, which ultimately form proteins. Functions of proteins. Proteins have many important functions in the body. Structural proteins provide support and strength to tissues, such as collagen. Enzymatic proteins catalyze biochemical reactions, such as amylase. Transport proteins move molecules throughout the body, like hemoglobin transporting oxygen. Regulatory proteins help control physiological processes, including hormones like insulin. Defensive proteins, such as antibodies, protect against infection. Motile proteins, including actin and myosin, are involved in movement. Protein solubility is affected by several environmental factors, including pH, ionic strength, temperature, organic solvents, chemicals, and heavy metals. When these conditions change, proteins may lose solubility and precipitate out of solution. One common method is salting out. In this process, high salt concentration competes with proteins for water molecules, reducing protein solubility and causing precipitation. Another method is isoelectric precipitation. At the isoelectric point, or PI, the protein has no net charge, so electrostatic repulsion is minimal. This allows proteins to aggregate and precipitate. Heat precipitation occurs when increased temperature denatures the protein, exposing hydrophobic regions. These unfolded proteins may aggregate and fall out of solution. Understanding protein solubility and precipitation is important in laboratory testing, protein purification, specimen handling, and interpretation of clinical results. Protein denaturation. Denaturation is the loss of a protein's native structure without breaking peptide bonds. It mainly affects secondary, tertiary, and quaternary structures. Denaturation can be caused by heat, extreme pH, chemicals, heavy metals, or physical stress. When proteins denature, they lose their normal biological function. Some denaturation processes are reversible, while others are irreversible. Methods of protein analysis. Several laboratory methods are used to analyze proteins. Electrophoresis separates proteins based on charge and size. Chromatography separates proteins according to their chemical interactions. Spectroscopy measures protein absorbance or fluorescence. Mass spectrometry identifies proteins based on mass-to-charge ratios. Immunoassays use specific antibodies to detect and quantify proteins. These techniques are important in research, diagnostics, and clinical laboratory medicine. Protein quality control. Cells contain quality control systems that help proteins fold correctly and function properly. Molecular chaperones assist proteins during the folding process and prevent aggregation. Misfolded or damaged proteins are often tagged with ubiquitin and degraded by the proteasome. These quality control systems are essential for maintaining cellular health and preventing disease. Summary and key takeaways. In summary, proteins are essential biomolecules with diverse structures and functions. They are composed of amino acids linked by peptide bonds and organized into multiple structural levels. Proteins perform structural, enzymatic, transport, regulatory, defensive, and motile functions. Understanding protein structure, denaturation, analysis, and quality control is fundamental in biochemistry and clinical laboratory science. These concepts form the foundation for many advanced topics in clinical chemistry and molecular diagnostics. Thank you for reviewing protein basics with Inanna Board Review. I hope this video helped simplify protein structure, function, and biochemical properties, and made these important concepts easier to understand and remember. If you found this review helpful, please support the channel by liking the video, subscribing, and sharing it with your classmates and colleagues. Also, don't forget to leave your feedback and suggestions in the comments below. Let me know what topic you would like to review next. Thank you for learning with Inanna Board Review. Understand, analyze, and excel.